Hi everyone, my name is Amir Mitchell, and today I'd like to talk with you about our work on bacterial adaptation to host-targeting chemotherapies and how that adaptation alters drug metabolism. Before I do that, I just want to say a few words about the microbiome, the human microbiome. The human microbiome is a collective term we use to describe diverse organisms, bacteria, archaea, fungi, and protists and viruses that pretty much cover every nook and surface on our body. We tend to think of ourselves as superorganisms because of the microbiome that actually contains about 2 to 3 times more cells than the cells in the human body. The microbiome brings to the table a huge pan-genome, lots of genes that complement our own genome and are essential for our health and development. In the recent decade or so, there has been huge interest in the microbiome and how the microbiome actually impacts our proper development and our health. Recent work also looks at how the microbiome interacts with cancer disease progression and treatment success. In very rare cases, we know of bacteria or microbiome involvement in causing cancers, but I would say that these are the rare cases. It is much more common, from what we know now, is for the microbiome to actually influence cancer disease progression through multiple mechanisms. Some of them could be interaction with the cancer cells or the immune response of the human host. Or another very frequent form of interaction is influence on treatment success by bacterial metabolism of drugs that are targeted at the tumor. So bacterial metabolism can have a huge impact by different forms of drug metabolism, either activating drugs, processing drugs, or degrading drugs on the treatment success. Recent work has shown that we need to think of the microbiome not only in natural microbiome sites like the gut or the skin, but actually also on the microbiome within tumors. We now know that a lot of tumors in different sites could become persistently infected with bacteria, and we term these bacteria that colonize tumors the tumor microbiome. In some cases, it's very natural to think of contaminated tumors, especially in sites that are naturally exposed to a microbiome, like the gut, colon cancers, mouth cancers, skin cancers. But we now know that cancers, even at very sterile sites, like brain cancers, bone cancers, pancreatic cancers, can become infected with a persistent tumor microbiome. So all the interactions I was referring to earlier, immunomodulation, drug processing and degradation, can actually transpire not only in natural sites, but also in the intimate settings of the tumor microenvironment. And these interactions can be very, very potent in that very specific niche. So again, all these interactions also happen within the context of the tumor microbiome. Another thing that is worth pointing out when we think about the tumor microbiome is recent attempts to use bacteria to intentionally infect tumors and colonize tumors as part of a therapeutic approach. There are now clinical trials trying to actually infect tumors specifically with probiotic strains or engineered strains that are supposed to either secrete toxins or process drugs as a therapeutic application. So again, that's a synthetic tumor microbiome, but it's also worth being aware of that context. Generally speaking, I think the field sees the microbiome as a modulator of the drug-tumor interactions. And the microbiome, either in natural sites or in the tumor, can implicate through diverse direct and indirect mechanisms the strength of that interaction between the tumors and the drugs. But I would argue here that this tumor-centric approach misses on something fundamental about chemotherapies, and that is that many host-targeted drugs, and definitely many chemotherapies, are highly toxic not just for the human host and for the tumor cells, but are actually also very toxic to the microbiome. We now think that about 50% of all chemotherapies are highly cytotoxic agents also for bacteria. So if we think about the same interaction from the perspective of the microbiome might need to actually adapt and respond to these chemotherapies with prolonged treatment. The questions I want to put forth here is, can bacteria, either within the natural sites of the microbiome or within the tumor microbiome, can evolve to become chemotherapy resistant? And if they do, will bacterial adaptation actually influence the host treatment itself? We know currently nothing about these processes, although they are very likely to happen. To study this hypothesis, we used a model system, a very simplified model system in which we used C. elegans, the worm, as our host, two fairly understood chemotherapies, 5-FU and FUDR, and in our case, the microbiome was actually the E. coli diet that the worms feed on and actually colonizes the worm gut. So this is our model system to try to understand bacterial adaptation and its host implication in the context of the worm host. Before I go into the research itself, I'll just briefly explain the drug mechanism or the toxicity mechanism of these chemotherapies. We have a fairly good understanding of the toxicity mechanism of these chemotherapies in bacteria and in host cells, in human cells and mammalian cells, and it's a very similar mechanism. So bacteria, like many other cells, can either uptake nucleotides and nucleosides from their environment using the salvage pathway. As you can see here in this example, it's uracil and uridine, or they can make their own nucleotides using the de novo nucleotide synthesis pathway. And of course, nucleotides are essential building components for the cells that are needed for RNA and DNA metabolism. The two drugs I was mentioning, 5-FU and FUDR, actually exploit the salvage pathway. They're transported into the cells using very similar mechanisms for nucleotide transport. They get phosphorylated, and they interfere through various mechanisms with RNA and DNA synthesis. Previous works have shown that in the context of a worm host, bacteria actually accumulate a toxic intermediate of these prodrugs, and that toxic intermediate, shown here in my diagram, is highly toxic for the worm host. Not only that, previous works have also demonstrated that mutations in the nucleotide synthesis pathway, either in the salvage pathway or in the de novo synthesis pathway in bacteria, can impact drug toxicity in the host. So again, this is just the mechanism of these two chemotherapies in bacteria and in the worm host. The first question we wanted to ask are these chemotherapies even really toxic for bacteria? In order to do that, we used standard sensitivity curves. We measured the bacterial optical density after 12 hours of growth in the presence of these two drugs, and we repeated our experiments both in nutrient-rich and in nutrient-poor media. In both cases, in both drugs, we saw that these drugs are indeed highly cytotoxic for bacteria. But one interesting observation was that the toxicity is actually much, much higher in nutrient-poor media. In retrospect, it makes a lot of sense. The probable reason for the high toxicity in nutrient-poor media is that nutrient-poor media does not have any nucleotides or nucleosides in the extracellular environment, and these potential metabolites can compete with these drugs for intracellular transport. So in poor nutrient media, there's no competition, only the drugs are transported into the cells, and that's why we see the higher toxicity, again, both for 5-FU and for FUDR. How do these toxicity profiles compare with what we see in cancer cells? Well, actually, if you compare it to previous works looking at the panel of a few hundred cancer cell lines, and you look at the median toxicity for these cell lines, you can see that the toxicity we see in the bacteria are comparable, if not even higher than what we observe for cancer cells. So we can definitely check that interaction. These two chemotherapies are highly cytotoxic for our microbiome, for E. coli. But what about resistance against these chemotherapies? Can bacteria quickly become resistant to these chemotherapies? We decided to first approach that question by performing a loss of function screen, genetic screen. What we decided to do was to map the drug resistome using a genetic screen approach. In these experiments, what we decided to do is to take a panel of 4,000 strains. Each strain is mutated in a single gene, and to test if any of these loss of functions actually confer resistance against these two chemotherapies. That allows us to map the drug resistome and see if bacteria can become resistant to these drugs with a single evolutionary step, with a single gene inactivation. When we did these experiments, we found about 100 different loss of function mutations that confer resistance. And what you can see here is actually clustering of these resistant strains. Red means high drug resistance. And on the x-axis, you can see which gene was actually inactivated. Of course, I don't want you to focus now on the gene names. I just want to make a very specific point, and that is that the resistome, the profiles or the different genetic mutations that confer resistance, seem to cluster not by drug type, but by media type. So we saw a very clear resistome for rich media, a very clear resistome for poor media, and a few mutations that confer resistance both in poor and in rich media. What's the underlying biology? So let's start off with the target pathway. In the upper right corner, you can see the diagram I showed you before of the drug toxicity. I just remind you that these drugs are transported into the cells and then are phosphorylated by the salvage pathway. And what we saw actually made a lot of sense. And that is that every mutation in the drug salvage pathway seems to confer resistance against these drugs, while mutations in the de novo synthesis arm of this pathway, of this network, confers drug resistance. But the remarkable thing was that the target mutations or the mutations were not specifically to the nucleotide synthesis pathway. So we definitely found loss of function mutations that confer resistance in that pathway, but we actually found many, many more mutations in other pathways. Looking into the biology, we rationalized that a lot of these mutations, either in central carbon metabolism or in secondary metabolic pathways, confer resistance by influencing the flux through the pentose pathway. The pentose pathway produces intermediates or precursor for de novo nucleotide synthesis. So what we reasoned is that you can confer resistance to these two drugs either by increasing input or flux into the pentose pathway by changes in central carbon metabolism, or you can reduce the demand for some of the intermediate metabolites of the pentose pathway, again by mutations in other pathways like the pathway for aromatic amino acid synthesis. So again, we now know that bacteria can become resistant very, very quickly by a single gene inactivation, and that could transpire through multiple different mutations in multiple pathways, not just in the target pathway. The question that, of course, arises is what will transpire under natural selection and what will actually be the host implication. So if bacteria do become resistant using one of these mechanisms, will that resistance have any impact on a treated host? In our case, on the worm. I think a good analogy to use is the famous sentence, all roads lead to Rome. In our case, yes, all adaptations will likely lead to increased drug resistance. The real question is, which road will bacteria actually use to get to the resistance, right? And will that road implicate or have any implications for host drug resistance? Now, of course, our genetic screen cannot answer that question. To do that, we can actually use lab evolution experiments where we gradually evolve resistance against these drugs under drug selection. That could help us understand whether evolutionary adaptation is random, each time taking a different path towards resistance, or do we see some convergence to specific mechanisms? It could also help us understand whether there are some forces that channel evolutionary adaptations towards some solutions and not others. And we can see whether we see mutations transpiring that are part of our loss of function genetic screens, or do we see completely new adaptation trajectories? So we did these lab evolution experiments. We serially transferred our bacteria in vitro for about 2 weeks in inhibitory drug concentrations, media containing inhibitory drug concentrations, and then we assayed our evolved strains. We tested if they evolved to become resistant. We tested their host implications, whether they impact drug toxicity in the host, and we also used whole genome sequencing to map the underlying mutations. And we did that in both drugs, both in 5-FU and FUDR. We also repeated that both in nutrient-poor and nutrient-rich media, and we did that with multiple replicants to see if we see the same, again, adaptations arising again and again and again in independently evolved replicates. In the end, we isolated pure clones, a single colony of the resistant strain, to test what are the underlying mutations. In all cases, we saw very high or very dramatic increase in drug resistance. That happened for both drugs in poor media and in rich media. And we also saw that in some cases, the increase in resistance was dramatic, almost 100-fold within a very short period of time. Again, pointing or suggesting that under the right selection, bacteria can evolve and become chemotherapy resistant very, very quickly. But the key question, or the interesting question for us, is whether these resistance at the bacteria level will have any impact or any influence on the toxicity of these drugs in a treated host feeding on the same bacteria. To answer that question, we used a co-culture assay where we co-culture the worms, our bacteria, either the ancestor strain or the evolved strain, and drug at different concentrations. We co-culture the bacteria and the worms and the drugs, and after 60 hours, we watch these worms under the microscope and we score their development. If there's no impact, there's no toxicity for these drugs, we expect to see full development. As you can see here in the black frame, we see full development after 60 to 72 hours. But if drug becomes toxic, we will expect to start to see delayed development of the worms, or in some cases, complete developmental arrest, and we won't see any worms, just the eggs. So that's our basic assay. It's not an assay we developed. It's an assay that was used quite frequently in the past in other papers. So how do our results look like? So what you can see here is just the ancestor strain, the ancestor bacterial strain being used as diet for worms. And you can see that at least in the context of FUDR, one of our drugs, even very small drug concentrations impact the worm host, or the drug is still very toxic for the worms. And even with very low drug concentrations, we see delayed and later complete developmental arrest. So this drug is very toxic when our worms are feeding on the ancestor host. When we fed our worms on one of our evolved strains, we actually saw very reduced drug toxicity. So again, the same worms, the worms have never seen the drugs, they're just feeding on different bacteria. And in this case, the bacterial resistance actually confers some resistance to the worms feeding on these bacteria. So you can see here there are many more black frames, meaning we see proper worm development at higher drug concentrations than we saw for the same worms feeding on the ancestor strain. And of course, you can see we can call this reduced drug efficacy. We can look at other strains, and again, we have many evolved strains, and we can score them for either reduced efficacy or unchanged drug efficacy. But of course, we don't want to stare at hundreds of different images of worms. We can summarize this in this bar graph. What we see in this graph is a summary of the impact our evolved strains have on worm development. On the x-axis, you can see all the diets we used, either the ancestor strain on the leftmost bar and the evolved strains on the rest of the bars. And the y-axis actually scores the worm development, the host development at different drug concentrations. In this case, we're looking at 5-FU. And I think what you can see very nicely is that the host impact can be grouped into two groups. There are some bacterial diets that go on unaffecting the host drug resistance or drug toxicity. And there are a few of our evolved strains that have a very, very clear impact and reduce the drug toxicity in the host or increase the drug resistance in the host. These are the rightmost bars in our graph. The remarkable thing we saw was that these actually group very, very nicely with the evolutionary history of the evolved strains. Again, in all cases, bacteria evolved to become drug resistant. But only bacteria that evolved in poor nutrient media evolved to become resistant, but also conferred that resistant to the worm host. Bacteria that evolved in rich media became drug resistant themselves, but that mechanism of resistance had no impact on the worm host. A similar picture emerges with bacteria evolving resistance against FUDR. Only bacteria that evolved in poor media conditions evolved to become resistant, but also had an impact when they were used as diet for the worm host. While bacteria that evolved on rich media conditions became resistant to the drug, but the resistant mechanism had no impact on worm drug toxicity. Going back to the analogy of all roads lead to Rome, yes, in all cases we saw increased resistance, but it seems that evolutionary adaptation is not completely random, it's convergent. All strains that evolved independently on rich media conditions had the same impact on the worm host. They had no impact on the worm host. All strains that evolved on poor media conditions reduced drug toxicity in the worm host. What are the forces that channel evolution? They seem to be the nutrient availability during the evolutionary process, during the evolution period. And what is the underlying mechanism of action? Is it a new adaptation or one of these loss of function mutations we already saw in our genetic screen? We can't answer right now, but we can definitely sequence the genome of the evolved strains and try to poke and get an answer for that question. What you can see here is the results of the whole genome sequencing of these evolved bacteria. The concentric circles actually show the chromosomes of 4 independent lines that evolved in different media conditions in the 2 drugs. And the points you see in these concentric circles indicate that we annotated a mutation, either a loss of function or potentially a gene-modifying mutation. And of course, it's very hard to stare at these. Let's summarize this in a table. So what you can see here is a summary table of which genes are hit again and again and again in these evolution experiments. That's on the x-axis of this table. And on the y-axis, you can see the evolutionary history during the evolutionary period. You can see that there are some genes that are hit again and again and again. Genes that are shown in bold are genes in the target pathways. So we can definitely see some mutations in the target pathway for nucleotide synthesis, but we can also see mutations in other pathways. Another interesting thing to observe is that mutation seems to be clustered by the media type during the evolution experiment and not by the drug. So we can find media, poor media specific mutations in UPP and PYRE, while in rich media conditions we see other mutations altogether in FIME and PYRH. We also saw a few cases where we saw drug-specific mutations. I won't go into this. But again, mutations are just the tip of the icebergs. These are mutations in central genes for nucleotide synthesis, and they likely have a wider impact on the flux and the expression of other genes, right? The mutations probably have a much wider impact on gene regulation and genome-wide gene expression. To assay that, we actually used RNA-seq of the evolved strain and compared the gene expression we see in the evolved strains to the ancestor strain. Again, we don't have a lot of time, so I won't go into the details, but we definitely saw very, very far-reaching implications of these mutations to the gene regulatory network. And just to focus on the target pathway, we saw that in the evolved strains, we see reduced expression of the salvage pathway and compensatory increased mutations in de novo synthesis, again indicating that probably we have a balance of the two conferring resistance, less salvage and more flux through de novo synthesis of new nucleotides. So let's wrap it all into a single model. We start off with a system where bacterial metabolism actually leads to accumulation of a toxic intermediate for these chemotherapies, and that toxic intermediate is highly toxic for the worm host. In the naive state before any adaptation, both bacteria and the worms are very, very sensitive to these two drugs, to 5-FU and FUDR. If bacteria evolve in rich media conditions, they themselves become resistant, and they do that by reducing the flux through the salvage pathway, and by compensating by increased flux of the de novo synthesis pathway. But they still accumulate a lot of that toxic intermediate. Therefore, the drugs are still very toxic to the worm host. On the other hand, if the same bacteria evolve in poor media conditions, they become resistant, but they do that by completely inactivating the salvage pathway. They have no reason to keep that pathway active because there aren't any extracellular metabolites that they need to salvage. So they completely inactivate the salvage pathway, and again, they highly activate or they increase the flux of the de novo synthesis pathway. But that adaptation mechanism confers resistance both to them and also to the worm host feeding on these bacteria. So going back to our first triangular interaction, yes, in this case, we know that bacteria can evolve very quickly to become chemotherapy resistant, and that bacterial adaptation will frequently influence the host treatment. Think about that concept for a second. We typically think of evolved resistance against chemotherapies coming from genetic changes to the cancer cells themselves. But what we're showing here is that the same resistance could transpire by changes in the microbiome, either in natural sites or in the tumor microbiome, and not through genetic changes in the cancer cells themselves. Now, of course, this work is on a very simplified model system, the worm model system, but then my lab is trying to use other model systems as infected tumoroids or infected tumors in mice to study that adaptation process, and we're even looking at clinical isolates that we get from resected tumors from patients. Of course, each one of these model systems offers pros and cons when it comes to tractability and clinical relevance. The last slide I want to show you is how these experiments actually transpire in a mouse model system. What you can see on the left-hand side are luminescent bacteria that we engineer to be luminescent. If we inject them to the blood system systemically of mice bearing subcutaneous tumor, they very quickly home and stay specifically within the tumors. And they do that for a long period of time. So they do home the tumor and they do form their own microbiome there. And then we can resect these tumors. That's what you can see on the right-hand panel. It's a resected tumor that still has these live bacteria. We can isolate these bacteria, sequence them, and study them. I'll wrap up by thanking the people who were involved in this research, my two fabulous students, Brittany and Serkan, Peter, who was a rotation student in my lab during the period where we did this research. This research was also a collaboration with Marion Walhout's lab and her student, Ari, from the Department of System Biology, and of course, my funding sources from the NIH. Thank you very much, and that will be it.